also early in the days of phage hunting, and he took on a master's project, one of those interdisciplinary Masters of Science projects that we have, as we don't offer a biology degree, and I think it's an extraordinarily flexible option for us. But he compared Maddie and slobber phages, from C.R. Anderson Middle School.
Slobber I found myself.
Yeah, thanks for the invite to talk today. My talk today is going to talk about how the work on phage kind of directly led to some of the things I look at today. I was a very early phage digger. I grew up in Montana in Great Falls, and by Montana standards, that made me a city kid, where most of my cousins are farmers up in the Shelby, all the way out to far eastern Montana. And it was really, within my own family, you can really see the stark differences in educational opportunities across the state. And so it's really great to see how this program has expanded and providing those opportunities to students. My mom actually grew up near Shelby. She was born when my grandparents lived in a camp trailer on someone else's farm. And she went to one-room schools and was bussed all over northern Montana as they moved around. And so I've really benefited, kind of as those educational opportunities have expanded in Montana, and I can see the phage digging program making a big difference in students, especially as many of you guys have presented. So I initially went to Montana Tech. So I was always interested in science through high school, had some great teachers in Great Falls. But Montana Tech was the first place I actually got to do science. And so I actually first started out in engineering and made a mess of that and came scrambling to the Dr. Douglas in the biology department. And he gave me an opportunity to kind of a second chance into the biology program. And then gave me an opportunity to work first with the hantavirus program, working with Amy Kinsey and Arlene there. And that was the first time I got, well, what's a virus, and got understanding what that is. And I really wanted to dive more into that. And then I took molecular biology from Marissa, and that was the first time that I really got exposed to molecular biology and really started like, okay, this is really what I want to do. You can talk about kids first holding that pipette. Like, I still am like, now I'm in my office writing grants, and I'm like, I want to get back in the lab and really do the experiments. And key experience I got with that, it was the first time I'd ever cultured a bacteria, and any time I'd actually infected anything. And those are still like daily things that we do in the lab today. And the first time I got into genomics, this has been a very key to my current research going forward. We're using it in very different ways, but genomics. And then a big part, the one that actually literally pays off now, is writing. The first like scientific writing course I took was taught by Marissa, and I still have some of the lessons kind of echoing in my head of like data are plural as I'm preparing manuscripts and grants. And then her mentorship style really paid off. And so her idea, her work with peer mentorship. So I wasn't in the lab very long before I was thrown in with other scientists in the lab, ranging from 10-year-olds up to Sandy Wardell, who I learned a lot from, and worked with them. I got to participate in a number of different outreach visits and gear up camps. And I was only there for a year and a half maybe, working on that master's program. One thing I wasn't good at is taking any pictures, or any ones that I did take died with a laptop that I had back then. But here's an alignment diagram of the few phages we had sequenced in the lab at the time. And so I sequenced Maddie and slobber and did the genome annotations for those, and worked with those and did the lysogens and a lot of that kind of stuff. And I'll highlight some aspects where this has popped up in my current research. So since the phage program, I went down to Texas A&M and I did my PhD with Dr. Benedict. Marissa was instrumental in helping me apply for grad school because I had no idea how to apply for grad school or what those programs were. And there I worked with another phage biologist, so he cut his teeth on lambda phage, is one of the classic E. coli phages. And we used a lot of the recombinatory tools that have been developed from phage to engineer cyanide degrading enzymes with the goal of degrading mine waste. So inspired by my time at Tech, worldwide cyanide leach mining is still a primary method for extracting gold and other minerals from ore, but it's really dangerous. If it leaks into a river, it sterilizes it with a matter of minutes. It's very toxic. But I'm not going to describe this today because it's a lot of enzyme kinetics, and I don't even like looking at enzyme kinetics. But during that, I still maintained that interest in infection. And one of the things that mycobacteria can do, and many other bacteria, is it can actually behave like a virus in some ways and infect eukaryotic cells and survive intracellularly. And I got more and more interested in that through journal clubs. And so if any of your students have opportunity to participate in journal clubs, I highly recommend those. And through that, I became aware of Dr. O'Connor's work, and I joined the Johns Hopkins School of Medicine as a postdoc in her lab. And there I looked at how amoeba drive the evolution of intracellular pathogenesis using the model bacteria, Legionella. And then while I was finishing up my postdoc there, my wife was like, "Have you heard of Washington State University?" And I was like, "Yeah, I think Marissa went there for a postdoc." And so while I was finishing up my postdoc, she moved to Washington State University, setting up her lab looking at tick-borne disease and the immune system of ticks. And so I kind of bounced back and forth a little bit and got to work on some other intracellular pathogens for a year. Francisella, it's a biosafety level 3 pathogen. And then while there, I discovered Anaplasma phagocytophilum. And it has a lot of similarities to Legionella. And so I was given the opportunity to take what I learned in Legionella and then try to apply it to phagocytophilum. And so I've kind of worked my way up through that, through grant funding and some opportunities provided by our department head, Kelly Brayton. And now I have my own lab. I was promoted to faculty in 2020. I tried to get connected with teaching in the phage program there. And then right as I was getting my foot in the door and started getting to know them, 2020 hit and we had to shut down teaching and the microbiology lab teaching. And so I focused on writing grants, and now I'm looking at how anaplasma adapts between the mammal and the tick. And so I'll be talking about these projects primarily, my work with Legionella and then with anaplasma, because I think both of those projects are directly inspired by some of the work on phage. So first, tell you, well, what is Legionella? And so Legionella gets its name from a conference of Legionnaires where there was a mass infection at a conference in a hotel, and these were all World War veterans, and they heart of the Cold War, and they came down with a terrible respiratory disease. And they thought it was a bioterrorism attack. They thought it was the Russians. And so it really led to a giant expansion in what we know as the CDC today. And what they figured out, it was actually the air conditioning system. So this bacteria, it can contaminate just tap water even. And if chlorine levels drop, it can able to expand in tap water. And in large buildings, they use water to cool the air conditioning system. So you can see here this mist coming off of a cooling tower in a large building. And it turns out that was what was spreading this bacteria and infecting people. In the lungs, it can infect our alveolar macrophages and survive within our macrophages in a similar way that things like Mycobacterium tuberculosis can survive in macrophages. A few thousand cases per year, but it's largely underdiagnosed because it can infect, it primarily shows up in elderly and immunocompromised, like World War II veterans, even in the 70s. But in healthy people, we get a lot of cases as well, but it's self-clearing. So those are usually referred to as Pontiac fever. Apart from cooling towers, it's also can be spread by potable water systems. So in water systems where there's not enough chlorination, you can have it accumulate. And also fun things like hot tubs and hot springs after you guys have been soaking out there. I'm sure Fairmont does a lot of work to keep their systems clean. But it's usually in like backyard hot tubs. Or there was a large outbreak at the Playboy Mansion one year. The bunnies were fine, but the old guys that were there got sick. And this is an environmentally acquired pathogen. So there's no vertical transmission from person to person. It's always an accidental exposure from the environment. So these bacteria don't want to be human pathogens. We're a dead-end host for them. So why are they able to infect us? So their environmental reservoirs are amoeba, so phagocytic protists. And so what are amoeba? So these are free-living eukaryotic cells. And so you can see here some different shapes, but they all have this amoeboid shape. But they even look fairly different. So this is an eyeball-eating amoeba. So change your contacts. Always use commercially sterilized contact solution and never sleep or swim in lakes with contacts. Down here is everyone's favorite brain-eating amoeba that people encounter in hot springs. Usually primitive hot springs, not developed hot springs. But yeah, if it gets up into your sinuses, it can follow the nerves up into your brain. But Legionella is happy to grow inside of these amoeba. So the amoeba generally are bacteriovorous. They're going around eating bacteria in biofilms. But if they encounter Legionella blocks digestion and it tricks the cell into forming this replicative vacuole and taking on a virus-like life cycle. They can also exacerbate the bacteria in water systems because these infected cells can also form cysts, and those cysts can prevent sterilization through chlorine or disinfection through chlorine and things like that. And these amoeba are actually very diverse. And so we often focus on animals and plants, and occasionally people look at fungi, but the bulk of eukaryotic diversity, multiple kingdoms of eukaryotic diversity, are all protists. And so these are single-celled eukaryotes, and they're more diverse than we are from plants. And among those, you can see many of them have amoeboid shapes. So you have the true amoebozoans up here. They're all amoebas. But you also have amoebas popping up, or amoeboid cell types popping up in other kingdoms, and including our immune system. And so many of the cells in our immune system are bacteriovorous, amoeboid-like cells. And so the idea is that these amoeboid protists may act as a training ground for pathogenesis. And indeed, Legionella can infect members we know it infects human cells, amoebozoans. It can also infect some bacteriovorous ciliates, and then these heterolobosian, these are the brain-eating amoeba, aren't actually amoeba, they're something weird. Not much is known about these, besides when they happen to pop up as weird pathogens. And so to look at, Of course, I loaded the wrong version. Another interesting thing about these is when I first started getting these in the lab, a lot of them don't like recovering from the cyst stage. To coax them out of the cyst stage, you feed them on bacteria before you then clean them up and try to get them growing exenically. And to do that, you do plaque assays. And so you can actually grow these amoeba on lawns of bacteria. And if you plate them out right, like we would do with the phage, you can actually purify plaques. And so that was how I actually recovered some of these initially from culture was, top or plating of a bacterial lawn. So to look at how Legionella is surviving in these different amoeba, I then went back to the genomics. And genomics had come a long way while I was in grad school. And not only can you identify what's there, but you can quantify things using genomics. And so I combined that with a large transposon mutant library. So each mutant in a library of bacteria has a mutation in a different location on the chromosome. And we were able to grow those libraries and identify what mutants are there and then quantify how abundant each mutant is used by how many times it's sequenced by high throughput sequencing. Then we can take that library and put it in different amoeba and then recover that after growth in the amoeba and resequence it. So mutations that disable its ability to grow in the amoeba will either be depleted in the output, whereas things that grow just fine will either not change or actually go up. And so for that, I can get a fitness score from every mutant in the genome. So we had, Legionella has about 3,000 genes, and we had over 8,000 mutants. And so we're able to hit every gene at least a few times. Anything that's essential for growth in media dies right away, but we can specifically look at things that are important for growth intracellularly. So we spread out those fitness scores. So here is how those fitness scores down into the things that are important for growth, and then spread that out by statistics. We can see out here in this left wing of this volcano plot are genes that were important for growth in, I think this was acanthamoeba data. And highlighted here in blue and orange are components of a type 4 secretion system and proteins that it secretes. So this actually looks like a phage tail. And so it's a type 4 secretion system, and bacteria can use these to secrete not only DNA, but also, in the case of Legionella, these effector proteins. So Legionella secretes over 300 different proteins that go in and reprogram the host cell, inactivating the immune system, reprogramming the cytoskeleton of the cell, redirecting vesicle trafficking, all to feed and benefit the bacteria. And so it's this really complicated virus-like interaction with the cell. So after setting some cutoffs, we can see that there's 200 and something different genes that are important for growth in each of these amoeba. So those cutoffs in kind of these red regions here. But interestingly, these genes that are important in each of these amoeba aren't necessarily the same genes. So if we look at the overlap among these, we found that Legionella uses different combinations of genes for infecting each of these different amoebas. There's a core set that are important in all of them, so the type 4 secretion system itself and core metabolic genes and things like that. But many, especially the different secreted effectors and other pathways, were important for growth in the individual amoebas. And we did a lot more complicated analysis on this and worked with a data scientist to sort all this out, which is described in our paper, this QR code. But the big takeaway from that is we did the same analysis during growth in human macrophages. And so we did primary bone marrow-derived macrophages from mice and also some human cell lines. And what you can see is the overlap of the genes important in here in Acanthamoeba castellanii overlaps with the genes important for growth in macrophages. But then if we look at the overlap of these different sets, we find is that each amoeba looks like it's selecting for different combinations of genes that together enable these bacteria to grow in macrophages. And so it emerged that our model is that Legionella has evolved this ability to infect macrophages by this expanded host range, by collecting genes important for infecting these different amoeba and still unknown protists in its host range. It's accidentally stumbled upon the ability to infect macrophages. And we think this extends far beyond just Legionella, because many other human pathogens have been shown to be either resistant to amoebal grazing. A lot of these are the mycobacteria here. Apart from Legionella, mycobacteria are the most common amoeba-associated pathogens in water systems. And further, even things like chlamydia and other very specific human pathogens, we find relatives of them in the environment with this relationship with amoeba. The work at Johns Hopkins was in the Tamara O'Connor lab. My co-author, Soma Ghosh, she did a lot of the data analysis, helped me with the computational aspects of those, and other diverse members of the lab and collaborators that provided materials and funding from the NIH. So while I was finishing up this work was when I made that transition to Washington State University, and we made the drive in a Honda Civic with bald tires across North Dakota in the middle of a snowstorm. And Dana was from California, from the city, and so I was like, we're driving through Montana. She's like, what? Can't we fly? And so we moved to Washington State University, and there I got interested in anaplasma and anaplasmosis. And so it has a lot of things in common with Legionella, and it's wide open. It's really unexplored in a lot of ways. That's because it's, unlike Legionella, we can't grow it on a plate. It's obligate intracellular pathogen. And you can see it here, all these little specks inside of these cells are bacteria, and they form these little berry-like clusters inside of the cells. And this is how we monitor it. Since we can't grow it on a plate and count it, we have to stain for it and visualize it by microscopy. It's a tick-borne disease, and so instead of jumping between all these different amoebas and humans, it goes between mammals and ticks, back and forth. And so simplified my system a little bit one way and overcomplicated it, and then we can't do genetics very well. It has really vague symptoms, and so fever, chills, Pepto-Bismol symptoms, muscle aches, and this overlaps with most of the other tick-borne diseases. And so things like Lyme disease have these very vague symptoms, really hard to diagnose. And you can have this instead or on top of those other tick-borne diseases. Unlike some of them, anaplasma can actually be fatal if untreated, and so it's important to figure out what disease you have. Luckily, antibiotics, broad-spectrum antibiotics, can cover a lot of tick-borne diseases at the same time, assuming you haven't gotten some weird resistant strain. But anaplasma, it's spread by Ixodes scapularis and Ixodes pacificus, so the black-legged ticks and West Coast black-legged ticks. And climate change has really been driving the expansion of the range of these ticks, and we're seeing anaplasmosis and other tick-borne diseases pushing up into Canada and infecting areas of the country we hadn't seen them before. And we're seeing more and more cases on the West Coast now. The ecology of those ticks is a little bit different out here, and so it's less likely to get it here. Luckily, there's not too much in Montana because of the drier climate. These ticks are more resistant to desiccation. We still have other tick-borne diseases that are related to anaplasma, so other rickettsial diseases in the Rocky Mountain West, so things like Rocky Mountain spotted fever. But anaplasmosis, those ticks need a drier environment. And anaplasmosis, or anaplasma, is really tied to the tick cycle, infection cycle. So there's no vertical human-to-human or mammal-to-mammal transmission. And there's also no tick-to-tick transmission directly. And so this bacteria has to go back and forth between the tick and a mammal. Sometimes some birds can be infected as well. And these ticks feed once per life stage. So the larvae acquire a blood meal from usually a small mammal, and that's where they pick up the anaplasma. And then those ticks digest their blood meal, and either in the next season or even the next year, they emerge as a nymph, and those ticks feed again. And that's usually where you get transmission to humans and livestock, is by the nymphs. They can also be transmitted again in the adults, and the males will feed a little bit more than the females. So the males will hop from animal to animal looking for mates, and you get a lot of your cattle transmission of anaplasma is through that mechanism. In both the mammal and the tick, this is an obligate intracellular bacteria. It must be inside of a host cell to survive. And like Legionella, it stays in a membrane-bound vacuole, blocks digestion from the cell, and reprograms the cell to redirect material to this vacuole to feed the growing bacteria. However, we know much less about this intracellular life cycle than we do in Legionella. And further, it has to carry out this life cycle in both the tick and the mammal. What we know is, well, ticks and mammals aren't the same thing. So they're separated by over 740 million years of evolution, very different organisms. And they actually, anaplasma evolved with these organisms. And so its closest relative is actually Wolbachia. And so it evolved from these arthropod-associated bacteria. In the mammal and the tick, it affects very different tissues. And so in humans, Anaplasma phagocytophilum has the weird aspect. It actually infects our neutrophils. So the most toxic immune cells in our body, it infects them, pacifies them, extends their lifespan, and replicates there. Other anaplasma can infect red blood cells, different tissue types, macrophages. But in the tick, it infects the midgut and then makes its way to the salivary glands and hangs out in the salivary glands waiting for that next blood meal. Arthropods and mammals both have immune systems. So mammals have an adaptive and innate immune system, but arthropods also have their own immune system. And that's what my wife, Dana Shaw, studies, is the immune system of the arthropod. In response to these differences, Anaplasma reprograms 43%, over almost 44% of its genome during mammalian or tick infection. And so there's a massive transcriptional shift, and we can use genomics to quantify those transcriptional differences. Many of the genes that are highly expressed in the tick, we have no idea what they do. So most of those tick-specific genes are uncharacterized, and we don't know what they do. And then further, many of the genes that are upregulated during tick cell infection, we can knock them out through random mutagenesis, and they can still grow in the mammalian cells. So I was curious, well, and I loaded the wrong version again. So I actually looked back at our old phage genome annotation, and we annotated the repressor in slobber and matty. And so one of the ways that phage make their decision between whether they're going to go lysogenic or the lytic cycle, or if they're expressing early genes versus late genes or the replication, are through transcriptional regulators. And depending on what DNA they bind and how, they can turn on and turn off different genes. So I looked in Anaplasma, can I identify any transcriptional regulators? And the highest expressed gene during tick cell infection was this TR1, with transcriptional regulator 1. And it actually looks like a lambda phage transcriptional regulator. And so it has this helix-turn-helix dimeric structure. So this is from AlphaFold. And it looks just like a phage regulator that makes the decision between lysogenic and lysogeny. And here you can even overlay our predicted structure with a known structure of a helix-turn-helix regulator superimposed onto DNA. And so, okay, we think we have a regulator. Well, is it actually important for that switch? So we went to a random mutagenesis library, and with a lot of screening, we were able to isolate a transposon mutant in TR1. It grows just fine in mammalian cells, but when we put it in tick cells, it's unable to survive. And so it steadily declines. Growth in tick cells is real slow, but our control strain is able to grow just fine. Similarly, if we put it into mammals, both strains are able to infect mice just fine. And then we feed ticks on these infected mice. The ticks are unable to acquire the TR1 strain from an infected mouse. So with this mutant, we've broken that transmission cycle. So these mice are the last thing that those bacteria are going to see. They're not able to complete the transmission cycle into the next generation. So if the transcriptional regulator, well, what is it actually regulating? So to look at that, we took time points early in this infection here to where before the bacteria had died off. And we asked, well, what genes are failing to express in this mutant? And we can see here is a lot of genes that are known to be expressed during tick cell infection, but they failed to express in the TR1 mutants. things that are likely to mediate host pathogen interactions, like membrane proteins, major surface proteins, other regulatory proteins, and then components and things secreted by the type 4 secretion system. Luckily, in our transposon mutant library, we had some mutants in some other genes, and some of those themselves are unable to grow a membrane protein and then a protein of unknown function. When those genes are disabled by the transposon, they too cannot grow in the tick cells. And then this 8A up here is what I'm continuing to work on. We'll talk about that in a sec. There's a lot of continuing to work on TR1. We don't know what it's actually binding, and so working with my collaborator Ian Cadby at the University of Bristol, and we're doing a lot of binding assays with purified protein, trying to figure out what DNA is it actually binding, and does that agree with our transcriptional changes to control for, make sure those aren't some indirect effects. And then coming from that, one of the main things that came out of that data was things we think might be secreted by the type 4 secretion system. So again, it's making a pilus, kind of like a long phage tail or filamentous phage that can secrete bacterial effectors into the host cell during infection. Legionella has over 300 of these. In anaplasma, we predict there's somewhere between 20 and 50, depending on which algorithm we use. But only 4 have been experimentally tested, and only in mammalian infection. So can we find some tick-specific effectors? And so we use some different machine learning algorithms, and in all cases, this gene that we've started calling, this protein we started calling 8A, for anaplasma tick effector A, was predicted. And it is specifically expressed during tick cell infection, but not during mammalian cell infection. You can also see this on Western blot. Skip over that. We test, there's no secretion assays available in anaplasma, so we used Legionella again to test if it can be secreted. So we fuse our effector to a cyclase and express it in Legionella, and then infect cells, and if these effector is secreted, it leads to the accumulation of cyclic AMP in the host cell, which we can measure by an assay. And in that case, 8A was significantly secreted during a Legionella infection. Similarly, we looked for mutants in this, and again, the mutant in 8A cannot grow in the tick cells. And again, there's at least a partial defect in the ability of ticks to acquire this mutant, indicating this effector is important for tick infection specifically. And then, well, if it's secreted into the host cell during infection, well, what is it actually doing in those host cells? And so I have a talented student, my first graduate student, who's going after this question. And so she expressed green fluorescent protein in eukaryotic cells, and you can just see it goes everywhere. And then when she fused GFP with 8A, 8A localizes to this webby network, and it's basically you focus just under the cell membrane. We see this web of material. And to us, this looked like it might be actin. And luckily, the first thing we stained for was actin. And we can see using a chemical called phalloidin, it stains the actin cytoskeleton. And so these long fibers, but also some dimmer network of cortical actin in the cell. And what we found is that 8A localizes very, very, very strongly to this cortical actin, so this webby actin. And it looks like it's also manipulating the phenotype of the actin fibers in these cells. And to this, it looks like cortical actin, so actin network, more mesh work of actin underneath the cell surface. And we can see if we stain for cortical actin, a binding protein, we see a similar localization pattern and quantified association with actin. So she's done a lot more work identifying what parts of the protein localizes with actin. And to get this cortical actin phenotype, it depends on multiple domains of the protein. And it has all these repeat regions, which we're currently under investigating to figure out what's going on. And so these results are summarized in our recent paper. But we're still doing a lot of work looking at 8A in more detail and how it's working together with other type 4 secreted effectors. We want to know, like, when to the infection cycle is actually important. Look at 8A in the context of infection, and we're currently working on more genetic tools for in anaplasma and in the tick cells to go after those. This is the old version. Sorry, the images are all messed up. I loaded the wrong version. But these are the members of my lab here. So Brittany Genera, Erica Warwick is our lab manager, who is reorganized the lab. I've never seen the lab more clean than a few days after she showed up. I've hired another scientist as a research faculty, Prabhat. I have 2 former interns that have gone on to bigger and better things. Kyle up here, and Daniel. So Daniel has a Fulbright scholarship. He's over in Germany right now. Kyle got his master's in education, not working with me, but at night without telling me. So I found out halfway through, and I'm like, what? That's great. And so he's working on a doctorate in education now at the University of Arkansas. And a big thank to the Brayton Lab. And so the Brayton Lab got me started working on anaplasma. And this is the lab with Brittany and myself covered up, and Deirdre hiding under there. And then the Shaw Lab, they were a big help on working on the ticks systems. My collaborators at the University of Bristol and other labs that have shared materials with us. And then our funding sources, and this is where those writing skills that Marissa taught me really literally pay off. Marissa knows how terrible my writing was when I was in the lab. And when I went to grad school, people recognized the Phage program, and they're like, oh, well, you're a grandchild of the Hatful Lab through Marissa. It's the idea that mentorship is passed down. And so Tanner and Brittany, who's covered up, would be Marissa's, grand grad students, and hopefully I can pass down some of her mentorship to them through their careers. So,
This is very awesome and very cool, and I hate ticks about as much as I hate snakes.
Yeah.
Probably worse. What is the thought of with the increased range, like treatment, diagnoses really? Like, what is the way to impact the reduction of the disease?
It's tough, and so a lot of trends are kind of exacerbating it. So more and more people are moving into environments that have the ticks. The ticks are spreading in their range. And so a big part of it's awareness and diagnosis,